Okay, so the next speaker is Max Schuback, and he's going to tell us about Mpre studies to identify disease variants, so kind of modern form of enhancer bashing.
Thank you for the kind introduction. Hello, everybody. Yeah, so I'm talking about regulatory variants and in enhancer and promoter region, or just call them now regulatory elements. So as you know, we have the human genome has mostly the non-coding part, and what's a bit of pity is if you go to databases and look if what variants are really known that cause a genetic like a Mendelian, a classical Mendelian disease. All of them that are identified are basically in the coding part, and there's only a few non-coding variants that can link to a Mendelian disease. So the question is what we need to find more of these non-coding variants that link to a disease. And I think what we need is a quantitative measurement of all these variants so to say something about the disease. And yeah, we can do this using massive parallel reporter assays. So with these Mpres, we can do three things. So first, we can test the activity of a region. Second, we can test different variants all over the genome. And the last thing, what we did in the study here is we have a really dense readout of a specific element. And again, these are 20 now regulatory elements. I call them promoter and enhancer because we did a slightly different Mpre here. So all of them are linked to a genetic disease, and most of them to cancer, but there are also all types of other diseases. So these are our selected regulatory elements. And so how our assay works, so first we try to integrate variants into our target region. So we used an aeroprobe PCR to integrate random variants into our target. Then we add a random tag. So we associate each of those random integrated variants a random tag to that. And finally, that's why I now call them promoter enhancer. We integrated into a plasmid construct, and we did a slightly different here. So in terms of an enhancer, we add a minimal promoter. So we have a core promoter, so we have some standard promoter activity for those elements. Then we express them into cell lines, which we think it's important for those elements. And finally, we extracted DNA and RNA out of it. And basically, we now just reading the counts, the tags, because they are associated to the variants. And so finally, we're having counts of DNA and RNA. It's basically how many DNA produces how many RNAs. So which variants produce which type of number of RNAs. And so here we are. So we have these counts, and all these tags are associated to variants, but to multiple variants, maybe 5 or 10 per fragment. So we have to filter them down or narrow them down to one variant. And here we use a linear fit model. So we use the log of the counts to get basically the log2 variant on the expression for every of those variants. And we have also some significance of the p-value, so we know we have the baseline, the wild-type activity, and basically this gives us how significant different is our variant in terms of the wild-type activity. All right, so I show you one example, and the example is TERT. So this is the telomerase reverse transcriptase, and this is a really important gene because it elongates the telomeres so that it's kind of can divide the cell. So this is an expression pro for how it looks like. And here we have on the x-axis all the possible variants. So from the transcription start is somewhere here and going in the 5' side. And on the y-axis, we have the log2 variant effect. So positive means we have a variant with an activating effect. Negative means we have a negative effect, a repressive effect of this variance, and 0 is basically our wild-type activity, so the reference sequence, basically. So what you can identify are these blocks here, and these blocks are mostly binding site domains. So if you kind of disrupt any of those positions in those domains, we see mostly a reduce in the expression effect, but also here, for example, this is a known ETF repressor site. Of course, they are also not only activators, they are also repressors. So here, this ETF repressor cannot bind efficiently to the sequence anymore if we change here the sequence, and so the activation can go up. So what we also see are these single peaks, mostly activating mutations, and those basically creating new transcription factor binding sites. And for TERT, this is really, really important because TERT is, so the promoter in TERT is known that it has frequent somatic mutations in cancers. And somatic mutations are mostly activating mutations, and if you then go to literature and look for different types of cancers of study, you see also the somatic mutations are enriched in those peaks. So we find all of them in these high activating mutations in different types of cancers. So this was an example how it is for TERT. So some general facts about our Mpre. We did these 20 elements. We had over 30,000 unique variants. So for the single nucleotide variants, we have nearly a full coverage of all of our elements. These error-prone PCR generates also one base pair deletions, but here it's not so frequent, so we can just say something about 25, so a quarter of all possible one base pair deletions in our assay. So here you can see the expression profile of all of these 20 elements. And basically what you can maybe see, you have this line, so the Y-trait expression, and it's super different between the elements. So in total, so in average, we have a 15% significant variant effects, but it's really different across all of these elements. So it's here ranges from 4 to around 50%. Mostly are repressive effects, but also around 37% are activating effects. And yeah, we see these, like I showed you before in TERT, these clusters, and these clusters we think are mostly binding site, where we have a block of significant effects. And yeah, of course, I think it's clear that deletion have larger effect sizes than single nucleotide variants because you disrupt the sequence more severe. What was really interesting is that, for example, for codons, so to going to the coding part, it is known that the codon uses is kind of robust in these frequent transitional changes. So you have the transitions that can happen much more often than the transversions, and we see the same thing for our regulatory elements. So transversion having larger effect size, so it's more robust to this frequent transitional changes, the regulatory elements. So my last part, I want to talk a bit more about functional scores. So there are a lot of scores out there, and to predict also non-coding variants. There's conservation you can use, of course. There's transcription factor motifs, and how well do they predict basically our 20 regulatory elements. So we looked into a lot of functional scores, just show you a few of them. So here we did a Spearman correlation. So this here's 0 basically on the x-axis, and so it goes up to 0.6 Spearman correlation, so it's not so well correlated. So here are the functional scores, and what you see basically, they all are pretty similar, like DeepC, Eigen, CAT, Linsight, and so on. There's no difference really between enhancers, promoters, only maybe DeepC works a bit better on promoters than enhancers, but in total, it's again, extremely different across the elements. So you have these white boxes here. Same for conservation. Conservation is also really similar to all these functional scores. And here we looked, for example, to Jasper, taking the best 10 percentile best matched motifs and doing a kind of a score here. And it's also pretty similar. So this means it's hard to tell which score to use, which one works best. So I'll show you 2 examples of 2 elements, what's really good for those scores and what does not really work well. So one example is the promoter LDLR. And here we have on top, again, the expression profile. It looks really similar to TERT. We have the blocks and the activating mutations here. We have the conservation scores, so fast cons here, different type mammals, primates, and vertebrates. And functional scores, here I plotted 4 of them, like cat or deep sea. And finally, I just went to ENCODE and ensemble regulatory build and just plotted what kind of motifs, what transcription factor motifs are known in this region. So what you can see is that in theory for LDLR, conservation aligns pretty well with our transcription factor binding blocks, but not in all cases. So like for these 2 positions that are marked here, we see no transcriptional changes. There may be a bit in the middle, but here there's basically no block really that we can see. So it's hard to tell if there's a conserved element side, if this is really important in terms of the for the regulatory element. The same thing happens for the functional scores. We have here the best correlation of all of the elements, so going up to 0.59, so 0.6 Spearman correlation. But basically, I have to tell you that they all of them using conservation scores in their integrative score. So basically, they do align well with the conservation. So they basically predict on conservation. So that's why they are pretty good in this case. And for motifs, some of them align well, like here with the blocks, but here in the middle, there seems to be some incomplete motifs now in the database. A totally different story is this enhancer sort 1. And here, this sort 1 is super conserved, but funnily not conserved in where we see the expression changes. So here, like in the middle, there's a block where it's not conserved at all. So we have basically a negative correlation here. The same happens with our functional score because basically they use conservation as one of their major predicting factors. And yeah, we have a negative correlation. And basically we have no correlation here at all for those elements. So how can we improve this? And so we use then, for example, some sequence-based approaches. So we went and downloaded the score for the delta SVM on the same, I think HepG2 was it, and look how delta SVM performs here. And it's totally different. It really performs quite well. So if you just get rid of all conservation, just training on sequence-based information on, I think it's DNase, then we have a much more better correlation of 0.5 than all of the other scores. And so it learns also some motifs that are not present, of course, in all the databases because they're really, again, incomplete. All right, so these functional scores here, there seem to be also for the elements, so this conservation is one of the major driver, but it does not work for all of these elements. Also for the functional score conservation they use, and I think what we really need is to integrate more of these sequence-based models. So like get K-MER SVM or convolution neural networks really trained on sequence information that we can improve the prediction. Because right now the integrative or the functional scores, they basically use all conservation and the prediction is for all of them pretty the same. So to conclude, I showed you this Mpre, these mutational scanning of these 20 regulatory elements. And I think from our findings, we think these Mpres are really a nice way to get quantitative readouts for all these elements. So we have now all these elements, and you can now look them up, your variant of interest, and see what kind of effect has this to the transcription. And yeah, these functional scores that I told you are doing not so well. They are likely insufficient, but I think now we have a data set that you can benchmark on. So if you develop a new score, for example, you can use the data. So we have a website here, mpre.gs.washington.edu. You can just download the scores and maybe benchmark on those, how well your score works. So finally, I want to thank all the people that are involved, like Martin, Jay, Nadav, and Zhenling, Fumitaka, and all the other guys. And especially you for your attention, and I'm happy to take your questions. Thank you.
So, great talk.
Thank you.
I was wondering what proportion of sites you found had that inverse conservation pattern?
So I showed you really the extreme example. So, and this is basically where we have the inverse. Mostly we have sometimes conserved elements where we do not see any changes at all. So mostly we see no conservation, but we see changes. But the inverse one, this one was really special, I think. I just cherry-picked that out.
Nice talk. So my question is, I was maybe not clear with this statement on the transitions versus transversions,
yes.
And then in your analysis with the motifs. So this analysis with the associations to motifs, take them all into account, or you have tried them separately as well, only transitions, only transversions, and seen a similar effect in the agreements?
So we looked for, and so transitional, these single nuclear changes, so we look for all the effect sizes for all transitions, and we looked the effect sizes for all transversion, and then compared how significant different they are, and that's kind of, the transversion have larger effect sizes, the variance, than the transitions.
But could it be, for example, that transversions could be more correlated to motif disruptions, and then you can see that?
There's already literature out there already looked into persistent weight matrices, and they just see the same thing there, that they have that changes in the persistent weight matrix transversion have larger effects than transitions. Yeah, so it's really correlated to the motifs. Yes.
You had one answer where conservation and activity was anti-correlated.
Yeah.
This is an N equal 1, but you have like more either an intuition why this is like this, or maybe some answers you don't want really to activate them, and that's why the conservation goes the other way around.
I think we have to kind of separate here a bit, like conservation with the activity, because maybe it's important in other tissues, or these elements come from somewhere else. It's something that's really somehow tissue specific. And I think that conservation is not the main driver, and it's maybe, yeah, I can't tell you why this is exactly in this element like this, but I think we have to really be careful if you're just going for conservation on regulatory elements. Yeah.
So at 6 o'clock, poster session has just